Hey again, everybody. Welcome to Q&A with A&V. I'm the V part, Vincent Racaniello, and with me is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. I am the A. How are you today?
I'm very well. I had a fever today because yesterday I got my second shot, and today I had a fever. How about that? Chills. Leo wants you to have a full spa day. I have to give you a full spa day.
A full spa day?
I don't know what that is.
I don't know. Yeah, I don't know what that is either. Why? When you need more details, I don't know what that means.
Someone asked about Rich Condit. He's okay. He slacked us today, and he's okay.
Great.
And is anyone interested in SARS-CoV-2 and COVID-19?
No, it's all about the weather. I read this whole article about like how the grid in Texas is like totally collapsed.
Yeah, they have big issues. Yep.
Yeah, and they're the only state that is separate from the rest of the union because they're deregulated. So it just goes to show you what Republicans do when they tell you that deregulation is great.
Okay, let's dive in.
All right.
So Kate wants to comment on us on the media going full tit booby on variants. Lori Garrett's is even more deadly, contagious, etc. I think go ahead, Amy. Let's hear your thoughts.
I think Lori Garrett might want to retract her article, if that's what it said. I didn't read the article, so I don't know what she wrote exactly. Do you?
No, but she's calling it more deadly, contagious, etc. You know, she's full tilt scare tactics.
Yeah, well, when she ponies up some data, then we'll discuss it. Until they pony up some data, I'm not going with that. I mean, it doesn't really seem that that's the right interpretation of the data. It seems to me that maybe the virus actually replicates a bit slower, and then it allows you to make more or to hide more or something, or it's more fit for whatever reason, but I have not seen where it causes an increase in deaths or hospitalizations. And contagious, I don't really know what that means.
These are more fit variants, and they can displace other variants. So that's how I look at Ray. The sky is not falling. No, that's pretty funny. Someone said, Vincent, how are you doing? I'm feeling fine. So I had fever and chills this morning, and I'm okay now. It's gone. Second dose. I'm all set. And I have 2 now stickers on my phone, right? Amy's kind of... Do you like that, Amy?
I do. I'm jealous. I didn't get any stickers.
Columbia, you want my stickers?
No.
So I got my shot in the same arm. I said I want it in the right arm. I don't think it matters.
No,
do you think it matters, Amy?
No, it doesn't matter. The immune response diffuses throughout the whole body. No, it doesn't matter. I wanted it in my left arm because I'm right-handed.
Do polyclonal antibodies work the same way as monoclonals? You want to answer that, Amy?
No, you can go first.
An antibody is an antibody. Polyclonal is your serum. It's got lots of individual specificity antibodies. You could look at your serum, your polyclonal serum, as a mixture of monoclonal antibodies. So they are basically working the same way. And monoclonals, we can make them and purify them and inject them into people, you know, give them 2 specific monoclonals, and that's it. So what do you think, Amy?
I believe I've argued that for when we've discussed about EV68 serology, right?
Yeah.
I've made that argument that a polyclonal solution is just more than one monoclonal in a tube. And so I mean, they're going to be, you're going to make more than one. So I don't know why it would be confusing. No, I don't think we're greater than anti-vaxxers. I like that. That's kind of
That's
cute.
good.
The math. That's
That's
math. We're greater than anti-vaxxers.
So Sam is sorry to talk politics. However, in response to Amy from last week, there are no vaccines given by Israel in the occupied territories.
Oh, that's not smart.
Even a donation of 1,000 Sputnik vaccines has to jump through a plethora of hoops to get into the world's largest prison, i.e. Gaza, and are being held at the border.
Still not smart, but I'm not in charge. I would have vaccinated. You're only as strong as your weakest link, and that is not good.
With the idea of broad-acting vaccines and antivirals possibly have unintended consequences, like wiping out some of the good viruses in our body. So, I mean, I think the broadly vaccines would be limited to, like, broadly neutralizing influenza vaccines or coronavirus vaccines. So that should not be an issue, right, Amy?
Right.
Now, antivirals, Amy, do you think we could ever get a pan-virus antiviral that would hit both DNA and RNA viruses?
No.
I don't think so either. But you could do all RNA and all DNA, right?
No.
You don't think so even then?
No.
So there you go. You really couldn't hit enough broad swath of viruses to make an impact on your virome, right?
I mean, we can just look at ribavirin. It crashes polio and picornas, has no effect on coronas because they have an exo.
Yeah.
So, no.
Freeze from Texas, San Antonio. Amy's correct. Texas is a mess.
Sorry. I feel bad. Somebody I talked to today, like their family is from 45 minutes outside of Austin or something or other. And he said that they have like 4 inches of snow. The whole family is like, melted down, and they're never going out until it's all gone. And they have enough. They've been canning for weeks for this, blah, blah, blah. And I was like, really? 4 inches? Okay.
These are the headlines that the general public cannot interpret. Study finds Kent variant may be 70% more deadly. This is nonsense. These are misused numbers, which are not really applicable. But the mainstream media likes to have a field day with this. That's how they get attention.
It's not just the numbers. More deadly. There's not an increased number of deaths from the variant.
They did a small study, and they found, looking at 8% of the data, that it looked like there was a slight increase, but they admitted, NervTag admitted they didn't have enough data. How many spike proteins does each virus particle carry? Should I get my spike, Amy, and count them?
Sure. Go get your glass Corona.
Well, this is a 3D printed one from Mark Martin. And
lovely. Looks like they're 60.
In the electron micrographs of coronaviruses, they're not as dense as influenza, but they're denser than HIV. So, I know that doesn't answer the question, but do you have any idea, Amy?
I don't know. I'll say about 30.
Really? Mm-hmm. 30 spikes.
Well, if they're not as dense as influenza, and there's like 5 GP120s for HIV, I go with 30.
Miguel says, "What about eye covering?" So this came up today. Actually, I was on a live stream, and healthcare workers may wear eye coverings. So in Daniel's place, North Shore, they wear eye coverings because they're in constant contact with infected patients all day. And under those conditions, the likelihood that you could get infected in the eye first is higher. But I think for us, it's more likely to go into our nose. That's what I think. Apparently, Peru is having a political scandal due to the secret vaccination of elites months before the vaccine was approved. Well, I'm sorry about that. The pandemic brings out...
I'm not surprised.
No, not at all. Do sequence changes in the variants ever interfere with PCR tests? Yeah,
Well,
so
that's a spike dropout.
the UK variant, there was a deletion which caused it not to work, and before they changed the PCR primers, they were looking at dropout as a measure of its frequency. So yes. All right, back to Jeffrey. Saying the variant is 70% more deadly isn't helpful. That's right. I think yes, you're absolutely right about that. I doubt the data to begin with. There are a few scientists say this, but for the most part, not everyone is agreeing with that. Not everyone is agreeing with the conclusions about transmissibility and so forth. Why does the S kept in an open prefusion improve the efficiency?
It allows for more of the epitopes that antibodies recognize to be available.
Yeah, when it goes into the postfusion, a lot of the epitopes are hidden. But Jeff McClellan, is his name? No. Jason. He said that it also improves the yield of the protein, keeping it in the prefusion conformation.
Yeah, but that's not the question. Question has to do with why the vaccine is better, works better.
No, but you can get more of it, though.
Yeah, but who cares? That doesn't have anything to do with it.
If hosts came first, then were viruses derived from rogue elements of the host? So the RNA world, which came first on Earth before cellular life, had self-replicating RNA elements. Eventually cells evolved, and then the RNA elements went into cells, stole capsid genes, and became viruses. That's one theory, and I like that a lot. Do you like that theory, Amy?
In part. Other parts I don't like.
Can I use my mic with a coronavirus on it?
Sure. Be my guest.
Here we go. How unusual is it for laboratory to sequence a virus but not make it publicly available, as is the case with RATG13? Well, they did make it publicly available. It was published previously. Actually, I think part of it was published. It was just a small bit, though. And then what, 100 bases maybe? What's a read, Amy, on high-throughput sequencing?
Depends on what platform you're using. It can be as short as 75. But I sequence viruses all the time. I don't make them publicly available.
Yeah, but I think now looking back on this one, so they had a very small piece, and it was in the freezer. And then when they blasted the SARS-CoV-2 sequence that picked up this one, and so they went back and did the whole genome, and then they published that. So I don't think there's anything nefarious there.
No, it's common standard procedure. But the truth of the matter is we sequence, everybody who's a virology lab basically sequences viruses routinely, and we never publish them. We never make them public. But actually, so I had a conversation with a colleague of ours this afternoon, and actually, RTG. So Lympha's Nature Communication paper, which surveyed bats throughout Thailand and Japan, so like a 4,300 square kilometer area between multiple latitudes, and he found multiple viruses in these bat populations that are more similar than RGT13. And actually, the spike of RG13 is less similar than some of these other viruses, and they don't use ACE2. And actually, RG13 was the weakest out of all of them in similarity.
I should point out that RATG13 has over 1,000 base differences from SARS-CoV-2, so it is not the precursor.
I'm just saying this idea that it came from a lab and all this other crap, just that one paper by that one survey discards that. Because if it came from a lab, you couldn't have all these viruses in a 4,300 kilometer square distance. That doesn't make any sense. You'd have one, and you'd never be able to find anything similar again. That's it.
Is there any peer-reviewed study demonstrating efficacy of monoclonal antibodies? Have you seen this, Amy? You're the one looking at
I think
the
Daniel has alluded to it. And I have seen some papers, but I can't remember off of the top of my head. And if I've seen them, then I sent them to Daniel, so he would be able to answer them because this is really his interest.
Yeah, there's certainly results out there, but I'm not sure the format. Daniel does talk about them a lot. Yes.
No, they've been peer reviewed. A lot of them have
Okay.
been peer reviewed.
All right, so here's an interesting one. If the vaccines are effective at preventing serious disease from the South African variant, wouldn't it be better not to develop a booster specifically because it will encourage an even stronger escape variant? No, the problem is if you have low antibody titers, that encourages escape variants because high titers will neutralize the virus and low titers will allow them to reproduce and you can select variants. Isn't that the logic, Amy?
Yeah, sounds about right.
Brits are saying their variant will take over the world. Not even during the British Empire days, their kings were that pedantic. Yeah. Well, it hasn't taken over California, for example. I think that's ridiculous to say it's going to take over the world.
Well, they're getting a lot of help. I believe it's the variant that the CDC came out and said would be like the predominant variant in the United States by March. Whether or not that's actually true. I mean, the truth of the matter is we don't do half as much sequencing. We don't even do like a tenth of the sequencing that we should be doing. So it's not really clear that you can make any conclusion whatsoever because you don't know what was there before. And I think like actually Biden's administration just promised like $25 billion to do more sequencing. Like to sequence 25% of all the patients who are positive. At this point, like you're gonna like if you believe the numbers that the pandemic, the number of people who are becoming infected is decreasing dramatically. 25% of that is you're still underreporting by like factors of 4 at least.
All right. If someone midway through their COVID infection gets monoclonals, recovers, doesn't get a COVID vaccine, does that mean they can still get infected by COVID again? Well, midway through.
They don't get infected by COVID. They get infected by SARS-CoV-2.
Yes, of course. And so when you get monoclonals, you suppress replication and you may not get a good immune response. It really depends on how late in the natural infection you got them. But, President Trump got, monoclonals, and so probably didn't develop his own antibody response. So I would say it depends on how long into the infection they gave you the monoclonals.
Actually, I would think that he did start to develop his own antibody response because it turns out that there was recent reports, I think over this past weekend, that he was far further into his course of disease than was initially told.
Yeah, that's right.
So that in reality, the monoclonals that he got didn't do anything. They did squat. So and when you test by the fact that like you gave them on Friday and your genius physician came out on Sunday or whatever the time frame was, but it was like 48 hours. Oh, look, he has an antibody response. You're just testing the antibodies that you gave him. It's not been 90 days. So you need to wait until like probably April to pull his blood and see if he has any response. But I think like what we can say is like I think that they didn't do anything. I think he probably... Well, you'll find out that he probably has a low level of antibodies, but whatever.
Why did the convalescent plasma people refuse my friend's regular donation once he'd had his COVID vaccine? That seems strange, doesn't it, Amy?
No. So the antibodies from his vaccine are solely against spike.
Ah, yes, that's right. Natural infection, many more, yeah.
Exactly.
This is Two Listening Ones New York Slice for today. We're the New York Slice, right, Amy?
Yes, we are.
Boy,
I'd like mine with black olives.
I would like a slice. I'll soon have both Moderna shots. Sometime later, when it won't deprive others, would a different vaccine add to immunity, for example, an inactivated virus? Oh, there are different proteins in an inactivated virus, right? Besides spike. Yeah. So that could help, yeah. Yep.
Yes.
Phil, I'm really enjoying the lectures. And Amy, your direct critical communication is awesome. Don't change that. Let me say something, Phil. She's not going to change. I've known her for 17 years. It ain't happening.
I think we've known each other longer.
Rich is in Texas where it's all messed up, but he said he's okay.
Good.
Can we expect a bump of cases due to huddling indoors? No, I don't think so. I think everyone's behaving properly, right?
Well, who are you huddling up indoors with during a snowstorm? They had to they had to already be in your house, right? So like Rich, is his grandkids with him, or they're in their own house and they got snowed into their own house and he's in his house with his wife, he's been there for like 100 years with.
Jeffrey says, I'm convinced many on Med Twitter are enjoying their 15 minutes of fame and secretly hope this pandemic continues. Yeah, people do enjoy their fame. Absolutely.
I am the one person who says, no need to make me famous. Isn't that me?
Amy doesn't want to be famous.
Hey.
Mark wants to know if Amy, this is for you, Amy. Do you think viral interference is possible with regards to flu and COVID?
No.
What if the influenza?
Similarity.
What if it induces interferon, and then that will inhibit the COVID replication? No, you don't like that?
No. I don't like that idea at all.
Okay. What's your position on mixing vaccines? You can mix Pfizer and Mrna. They're the same. We don't know if it works to mix them. So there are some trials being done to see if you can mix them. And I would wait for those results. I mentioned someone you know is previously infected and waited to get the vaccine. Did they eventually get it? I think this is Ian, right?
No, this is Nishay. And no, he has not. To my knowledge, he has not been vaccinated. He wanted, I think the agreement was that they would readdress in March. And it's not March yet.
I think I can guess, but what are your opinions on UK going ahead on a live challenge trial? I don't know why.
I saw that today in the Times.
It's ridiculous. What are they drinking? You don't need a challenge trial. You got plenty of cases. It's just crazy. This pandemic brings out the worst science in many ways.
Well, this pandemic has reinvented science. So, like, for instance, when you had Offit on, we talked about kids and why you would start off a pandemic with kids don't get sick. And his whole explanation was, well, you know, they didn't present symptoms, so blah, blah, blah. And it was like, did you not know that you're a pediatrician? How many diseases are kids just snot-nosed carriers of? Like, why would I not assume that a respiratory winter disease, some kid is not a snot-nosed carrier? Like, that didn't make any sense to me. So we've reinvented the wheel, and not in a good way.
Did you know, Amy, that CDC is now advising ordinary people not to wear N95 masks routinely? Is there any science behind this, or another empty piece of advice? No, CDC wouldn't do that, would they, Amy?
I take the fifth. I don't know anything about this. I
don't know what they're doing, and I see a lot of people wearing N95s.
My two students, Edmund and Michaela, my tech, wear N95s all the time.
Hey, Kimberly, Amy has relatives in Boca, right?
I do have relatives. My godson will be 15 tomorrow. I
heard a group working on a vaccine to target both S and nucleocapsid. What do you think? I think it's a good idea. What do you think, Amy?
It's great.
Yeah.
I think it's great. I heard the same thing. Yeah. Read this the other day.
How many SARS-CoV-2 particles are in Earth? So I heard someone say enough to fit in a can of Coke. I don't know how many that is. It's a lot of particles, isn't it?
Yeah, it is.
How many people are infected right now, Amy, on the planet, do you think? We could do the math.
Infected now? This
Right now, at
particular
this,
snapshot in time.
because that's what you want to know. We're going to calculate how many virus particles they contain.
Well, what I think it's thought to be like 2 million all at once. However, I don't think we have any accurate count for Africa. I think it's just.
Yeah, that's true. And we don't know how many
I don't think.
particles are
Yeah,
in each
I was
person.
gonna say I don't know how you would. I don't know how you do that.
I know
Without
you weigh. I can tell you there are 10 to the 16th HIV genomes on the planet because we know there are
How
about
can you know that?
35 million people. We know the average viral load. 10 to the 6th.
I don't know. I don't know that I would swear with that.
Okay, very Nika from Russia. Could you help make sense of the most popular anti-mask argument? If you're shedding virus, inhaling the contaminated air you just exhaled makes you more ill. That's ridiculous. If you're shedding virus, it doesn't matter if you re-inhale it, right, Amy?
Yeah, I don't understand that logic.
No, it's an anti-mask argument. It's ridiculous. Makes no sense.
Yeah, well, I don't get it. I think if you want to be an anti-masker, you need to come up with a better argument than that.
I'm sorry, I don't know anything about ITP. I had acute ITP as a child. Do you know what that is? No, someone will tell us.
Okay.
What do I make of the drop of cases worldwide? It's great news. It is a combination of, so many infected. And as Amy will tell you, there are many more people infected than we're diagnosing, right?
Oh, yeah, for sure.
So there are a lot more people infected.
I'm not sure I really believe that there's a drop, this significant drop that they're claiming that there is.
I think there is in the U.S., right?
I don't believe it, actually.
No? You think it's a testing drop?
Yeah, I think that people have decided. I think that there was an enormous amount of people who started at around, I don't know, late fall, who should have been tested, who thought that they were infected and refused to get tested because they were going to lose their job. And now I think that number has astronomically tripled because people say, there's a vaccine. I'm the next group. I don't need to go get tested. And all these, and you see the daily vaccine tracker on the Times, and I think we're almost at 5% of the population being fully vaccinated. So why do I need to get tested? And then people start talking about, first graders are going to be be able to be vaccinated by September. Now, I think the latest statement I saw this evening is that everybody who wants a vaccine could get one by April. So why do I need to go get tested? All these people are really vaccinated. So I will have hit herd immunity.
Well,
so I don't think that those numbers are reflective. I think those numbers are just reflective of the fact that people no longer go get tested.
All right. When the SARS genome is sequenced, is it only coding or the entire genome?
The whole genome.
Whole genome is, yeah. Oh, that's very nice. Super chat. Thank you. I forgot that I turned on super chat.
I don't know what super chat is.
Yeah, I turned it on, but I totally forgot about it. I'm so sorry. Super chat is when people can give you some money and their comment goes to the top. It just went to the top and disappeared. I don't know where it went. Thank you for the super chat.
Okay.
And thank you for liking TWIV. I made you fall in love with science. We could do nothing better than that. How close are we to true normalcy in the U.S. and throughout the world?
Well, Biden came out yesterday and said we'd be normal by Christmas.
I'll never be normal, Amy.
Well, you had to start with being normal to begin with, Vincent, to return to normal. You and I did not start at normal, so.
By the way, Everett is from Ukraine.
Good.
Should it be called this day in virology? Well, now we're doing super chat from Abraham. Can you artificially squeeze enough virus together? Oh, where did it go, folks? Where did the super chat go?
Here, can you artificially squeeze enough virus together to be viewable by the naked eye? No.
This day in virology, yeah, we're doing 3 times a week, but I think that's too much. We probably have to go back to, at some point, once a week. What do you think, Amy?
I'm sorry, what?
How many TWIVs do we need?
Vincent, I am finding you studio space with the idea that you have proposed that you wanted to expand the number of podcasts that you do.
Yeah, Amy's helping me find studio space in New York City.
Consequently, I am unclear what the answer you think you're. I'm unclear who you think you're answering, or who you think is at the other end of the telephone that is going to hear this answer. So I'm not. Yeah, I'm.
Right. Well, for now we're keeping it up, Amy. Claudia wants to know, how long does it take from entry of the virus in the body until antibodies disable it? Thanks, John Snyder, 2006, for your super chat. Thank you so much. Entry of virus until antibodies disable it. I think the infection, basically, if you, from the onset of symptoms, within 7 to 10 days, your virus titers are way down, and antibodies are not yet taking care of that. So, right, I think it takes antibodies disable it. I'm not sure they're disabling it in 7 to 10 days. But the antibodies take 2 to 3 weeks to build up.
Well, it takes 2 to 3 weeks to launch. It takes months to build up. I don't know why you people keep saying it takes 2 to 3 weeks to build up. You guys need to go back to Immunology 101.
You want me to show you my text? You want me to show you the curve in my textbook?
Well, I'm not sure your curve is correct.
Oh, well, that's not a good answer, Dr. Rosenfeld.
Well, let's just put it to you this way. I just wrote a grant on antibodies with an antibody person.
Okay. Let me show you the picture.
I know the picture. I've seen the picture.
What does it say?
It says that they start to go up, and then and then you can see IgG going up and plateauing all the way for months on end. IgM comes down, and IgA also IgA plateaus, but not to the same extent that IgG does. And I just wrote a grant that said, in the introduction, that upon virus infection and initiation or engagement of the naive B cell, it has to go to the lymph node to multiply and differentiate and blah, blah, blah. And that takes a minimum of 4 to 7 days. So I don't know where you people think you're coming up with this crap about, it takes 2 weeks.
That's when you first see the antibodies come on the scene. It doesn't mean that they're totally matured in 2 weeks.
Doesn't mean that they do anything in 2 weeks.
No, they're there. Here they are. Here.
They're there. Yeah, I don't want to look at this picture. Yeah.
See, there it is.
I see it. I've seen it before. And we've had the same discussion because we've had this discussion in terms of a certain individual. So I don't want to discuss it anymore.
When I get
you
to
guys
100,000,
keep forgetting that the immune response takes time. And I guarantee you that if you come out in 6 months, you'll see something different.
When I get to 100,000, YouTube will send me a play button award, I didn't know that. That's cool. I hope we're going to make it, I think, but it'll take a while.
Well, get your entire class to enroll.
Vaccines turn out not to slow transmission, then we would not achieve the true reach herd immunity that would protect the unvaccinated. If they do not slow transmission, that's correct. However, I'm thinking they probably will, but we'll see. Yes, you're right. Were all people who got the trial vaccines deliberately exposed? No, not at all. They're just sent back out into the world, and they went about their lives, and some of them got exposed, and those were used to determine the efficacy. They weren't deliberately exposed, no. Here we go. Regeneron paper. Studies in humanized mice and convalescent humans yield a SARS cocktail. I'm not sure that's an efficacy report, though. Amy, Israel is bound by the Oslo Accords, which puts the Palestinian Authority, not Israel, in charge of vaccination. The PH specifically declined Israel's vaccine offer.
Okay, well, whatever. It's still not, whoever made the decision, it's still not a smart decision. Okay.
First COVID challenge study has been approved to start in the UK. Yeah, I think that's crazy. And they didn't ask us. Nobody does, right, Amy?
No, they know better.
I love it when you interview Dr. Offit, but see if you can get him to speak more slowly. Yeah, he speaks quickly, but I think he can slow down the playback, right?
I don't know.
What do you think of the possibilities for future mRNA vaccine technology? I think it looks really good for other vaccines, and they're going to definitely try other viral vaccines. I know they have HIV and HCMV, cytomegalovirus mRNA vaccines in the pipeline. And I think it has therapeutic uses for delivering proteins.
They started off as making a cancer vaccine.
So I think we're going to see more of it because now it's worked, right? And it's proven principle.
Mm-hmm.
UK and California COVID variants found in Boise wastewater samples. Okay, not surprised. These viruses get around. We'll see what they do, right?
Mm-hmm. Mm-hmm.
Is it normal practice to use a vaccine that you're evaluating on the research staff? Not that I know of. Right, Amy?
I've never heard. I don't know. I didn't think that that's what they did. But, I mean, it's not normal practice, but I can't say that if you told me that Saban did that for his OPV, I would be surprised. I believe actually he might have.
What if hydroxychloroquine worked? You mean if it actually was an antiviral that you could take orally? And thank you, Tom Steinberg, for your super chat. Appreciate it. I think it could have made an impact on the outbreak if it were safe and efficacious, but it wasn't. But as Amy would say, it didn't work. Yeah.
I mean, I don't care about the rest. The fact is it didn't work.
Amy is a realist. All right, RTG 96 genome identity with SARS-CoV-2. When the actual bat host is found, what percent genome will be the same? What do you think, Amy? It's going to be 99.9.
What? Why? If I sequence the genome that was in my freezer at 96, and I go outside and I take another aliquot of RTG13,
no,
why
it's
do
a
I
different
think it's
when
going
the
to be
actual.
9?
No, I'm assuming it's a different virus for the precursor of SARS-CoV-2. That's what I'm assuming they're writing.
Oh, that's what they're writing?
I'm reading between the lines.
Well, I didn't get that from the question, but okay, sure. And I think Lympha's paper describes that.
So, thank you, Mary, for your super chat. Pair character doing a classic mic drop. Thank you. I have to figure out how to highlight these super chats because they come and go and I click on them and they don't do anything. Maybe somebody knows. What were we saying? Yes.
Lympha's paper, that's Nature Communication paper.
Yeah.
I think we'll highlight that.
Okay.
We'll highlight
We're doing that on Fridays.
Yeah,
too.
I mean, it's not just, he doesn't just look at bats. He looks at pangolins, coronas too.
300 watching, only 31 likes. Yeah, we have 412 watching. Do a like. Give it a thumbs up down below. Get us at least over 100. Is it true, as stated on a local public radio today, you can test for antibodies about 6 weeks after the second dose for proof the vaccine is working? Well, it's not proof that it's working. It's proof that you're getting antibodies. And you'd have to do the right test. You'd have to look for antibodies to spike, right?
Yes.
Does Since when do you believe anything the Washington or New York press prints? Their retractions have been staggering. Yeah, they have a big problem with science, in my opinion. I agree. Here we go. There's our slices, Amy.
Excellent.
Cheryl, true infection recreates many different antibodies to all parts of the virus. Vaccines only target spike. How many types of antibodies does a vaccine cause us to make compared to true infection? That's a good question.
So I don't understand the question, actually.
Well, when you get
I'm
spike,
a little
how many antibodies do you make? How many different antibodies? Thank you, Arthur, for your super chat.
Well, I think there are 20 epitopes, right? 20 total epitopes within spike, right? Or is that, or was Crotty and SETI saying that there were 20 epitopes all over the virus that they thought were
Spike.
neutralizing?
It was spike. That's correct.
Okay, so you have IgA, IgM, and IgG based antibodies, right? You have full class switching. Times 20. So you have 60 potential things, right?
Mm-hmm.
Not all of them are going to be, well, all of them in theory are going to be neutralizing. So all 60 of those will be neutralizing. And then who knows how many more you make that aren't neutralizing but bind or do nothing, right?
Yeah.
And then, I don't know, Ian's paper's not out yet, although I think it should be. They have a whole slew of other proteins that cause an antibody response.
Yeah.
Plus all the epitopes that are in nucleocapsid. So I don't know how to answer that question. How many different types? I don't know. Compared to a vaccine? Probably the spike-based only vaccine is, I don't know, 1/10 of the number that you would, if you had natural infection, you would compare. I don't know. What do you think?
Thanks for the super chat, Eugene. I think you have a restricted repertoire when you just do spike, obviously. And compared to a true infection, I think there are thousands in a true infection of specificities and much fewer, hundreds maybe, with the spike. Claudia, thank you for the super chat. Let's see if I click on those. Nothing happens if I click on the super chat. Look
so what
at
do
this.
you do with these $5 super chat stickers?
I give them to you.
Okay.
Amy looks like she's 17. She is.
Yeah.
She wishes, but she does look young. That's because I treat her well. Now she's going to get mad at me.
You send me to a spa.
I have to send you to a day spa. Penelope, can you state the efficacy of Pfizer, Moderna, and J&J? So Pfizer,
They're all
Moderna.
about 95, right?
Yeah, they're 94, 95. J&J was 72%. A little less. Is it a good idea to keep a snot rag in the freezer to be used later as a booster after one has beaten a cold? No, don't do that. No value to that whatsoever.
Oh my God, that's disgusting. Do you want your snot rag next to your ice cream? Who does these things?
Look, and we asked them to thumbs up, and we got to 162. That's great. You guys are great. Denise says, Amy, you are spot on. I would love to work in your lab with you.
Sure. Apply.
Amy's looking.
I'm always looking.
Will YouTube be in trouble after COVID? That's a great question. I suspect you guys will all be gone and you won't come to our live streams, and that's fine. No problem.
We're live streaming after COVID? This is news to me. Okay.
Well, live stream as long as people come. Right now, there are 442 people here. It's great. It's a community. And we made a community of people interested in viruses. And we could keep on chatting. What the heck? And, Amy, you don't have to come all the time. I understand.
Well, I think my virus is much more interesting. So, I like my virus. It's nice.
Here's a good one. Do you think kids should go back to school with current COVID positive rates of 8+% in New York City? I have a 5th and 8th grader. Is child inflammatory disease something to worry about?
I think you should handle that. You can feel the wrath of Theodore and Paul. I don't need to deal with it.
No, I think the positivity is too high right now to send kids back to school. Until they're vaccinated, you can't do that with that kind of positivity. And yes, the inflammatory disease is quite rare, according to Daniel. But
It's
you don't want
actually
to
been
happen.
reported to go up.
It's going up, yeah.
It was a big article. Yeah. All
Okay,
right,
Cheryl.
2
Yeah.
more questions and then I have to go.
Okay. This one's just a comment.
Offit is a must listen. Yeah, I think he had interesting stuff to say.
All right. So what's the current data regarding transmissibility in B117 as states are using this threat to slow our opening? I think it's absurd to use B117 as a threat to slow your opening. There is no evidence that this virus goes through the air any better than any other. It seems to be more fit and drives through the certain populations, but not all. As I've said before, it's in California and it's not displacing the current strain. So I think these policies based on these data out of the UK are wrong. But no one's asking us. Nobody's asking us.
All right, last one.
All right, let's pick a good one. Rich Condit's okay. Yep. If you are sick with COVID after the first shot, how long do you recommend before getting the second? You mean you get COVID after the first shot? That can happen, right, Amy?
For sure.
Yeah,
I know. My mom says to me all the time, "Oh, I heard somebody got COVID, got infected right after the vaccine. The vaccine must not be as good as you think, Amy." And then I ask, "Well, Mom, when did they come out positive?" And she says, "Oh, 2 days after they got the vaccine." And I throw up my hands and I say, "Really, Mom?"
Yeah. I mean, you have to fully recover before you get a shot. So whatever it takes, it could be a couple of weeks. It depends. But are you out of here, Amy?
I am. I'm going to go split some cells before I go home.
Thank you again,
All right.
Amy. Bye-bye.
Don't forget, bring your recorder, bring your mic, bring your
will.
testing equipment.
Bye.
Bye.
Okay. Now it's just me. I don't know if this is me. Is this me, Vinny? No, probably not. Are there cases of asymptomatic healthy individuals that develop lung damage? Thank you, Post Polio News, for your super chat. That's great. Now, I'm not aware of lung damage, but I am aware of healthy asymptomatics who develop long COVID. How concerned should we be about packages in mail or deliveries? Back in the beginning, we were concerned. I haven't seen any reports of transmission through that, so I think you're okay. Get the mail, wash your hands, be done with it. In Norway, 56 people died after vaccination. Yeah, I don't know what the story is there. All right. Something went on. We know this doesn't happen anywhere else. Thank you for the super chat, Tom. It's a pleasure to visit you. How do they know this virus can't infect birds and reptiles when they do not test them? You can look at ACE2 and say it wouldn't bind virus because it's too different. And you can be very sure of that. But you can take cells from birds and reptiles and test them. So you don't have to have actual animals if you wanted to do that. Are they still following the participants in the phase 3 to understand durability? Julie Wong, super chat. Thank you so much. I wish I could figure out how to, bring these super chats to the fore. Yes, they are following the participants in phase 3 for up to 2 years. Yes, they are doing that to see the durability for sure. Absolutely. Absolutely. Tracking infections for their MMR, does CDC solely rely on physician reporting? Clinical laboratories, mainly, send in the results of their tests, not just physicians. Yeah. Do we trust what the CDC says and recommends, or are they unreliable even with the new administration? I know a lot of great scientists at the CDC. I like them. I trust them. But the administration sometimes makes missteps. And, they're saying now that you could, if you're vaccinated, you don't have to quarantine. Thanks, Questella, for your super chat. So I don't trust everything blindly. No. And, people have accused me of being political about this. But I'm going to be critical of the current CDC, you bet. Are reports of idiopathic thrombocytopenia after Mrna vaccination valid? Daniel said this can happen on the last update. That's from him. Let's see. Are there other respiratory viruses that cause as many symptoms? You mean like these systemic inflammatory-based symptoms? So the other coronas, the SARS and the MERS, they can do this inflammatory disease for sure. Avian influenza viruses, when they infect people, they can cause inflammatory disease like this. So those come to mind immediately. Yep. Does the quality of your microbiome impact your ability to fight virus? Can bacteria eat virus? Well, no, they don't. But the microbiome influences immune responses. So, yeah, but exactly which microbiome, which bacteria, and how, we don't know yet. But people are working on that for sure. Oh, here's the super chat. Thank you. Now they're showing up in the feed. So I can say thank you so much for your super chat. I get how vaccines work in general, but how did they develop the mRNA vaccine? Well, they just said, look, first of all, DNA vaccines have been worked on for years. You take a piece of DNA that codes for a protein and you inject it into people. And it hasn't worked out so well. So they said, let's take the next step. In the cell, DNA goes to mRNA, to protein. So let's make mRNA. Pretty straightforward. I never thought it would work because RNA is quite unstable, but they wrap it in a lipid nanoparticle, and that really takes care of it. Okay, ITP is idiopathic thrombocytopenia, autoantibodies against platelets. Right. So the infection does seem to stimulate some autoantibody formation. So yes, that could be an issue in this person, and they should consult their physician for sure. If there isn't a true drop in cases, wouldn't the hospital still be overwhelmed? Excuse me. That's a great point. And Daniel has said that his hospital occupancy is down. So I think that the drop in cases is real, and Paul Offit thinks it was real, a combination of increasing herd immunity and vaccination. Moderna vaccines was developed within days of sequence publication. Couldn't challenge trials have gotten approval 6 months or more and saved 1 million lives? Back in January, February last year, we had no idea about the lethality of the virus. So I don't think we could have done that, knowing that, because people would have died in the challenge trial, because you need a control, right? Here we go. Abraham super chat. Yeah, can you squeeze enough virus together to be viewable? You couldn't see the individual particles. You could see a glop, but that would be a lot of viruses. Okay, so Tom, you found out about super chat. Thank you so much. Someone suggested I do it last time, and I did. Why did so many people die when this virus initially hit? Is it weaker now? No. What's happened is we're getting more population immunity, and that protects you, of course. And the virus first came in, everyone was susceptible, including older people who couldn't deal with it, and a lot of them died. So the virus has not changed as far as we know. Back to abnormal. That's great. I like that. I saw that dogs can be trained to smell out the virus. Yes, I saw that. I think they smell people who are sick, who are infected, and they give off a certain odor, right? They're not actually smelling the virus, but the volatile organics that you give off when you're ill. I don't think they would never be able to distinguish between variants. Fido can't do that. Oh, thank you, John, for your super chat. Purple Heart. So you think we need TWIV twice a week at least? Okay. I'm good with that. But I'm not sure Amy wants to do the live stream after the pandemic's over, but we'll see. Maybe I can get somebody else. We're going to do a live stream with Nell Zeldy in a couple of weeks. Stay tuned. Oh, here's a great one. When you look at viruses under a microscope, do they have colors, or is it just a convention that they're shown as gray and red? Totally a convention. No color at that low resolution, right? There's no light. The wavelength is too big. What does TWIV look like post SARS-CoV-2? We talk about other viruses, right? But we're going to talk about this one a while because there are a lot of interesting studies coming out. We're going to try and understand what went on and so forth. So, yeah, I think we'll keep it up for a while. The spikes are bigger and fatter at the end than I imagined. I should show you guys a picture of a SARS-CoV-2 model that someone sent to me today. Here, let me get this quickly. If I can find it, sometimes I can't find things quickly and I'm not ready. CoV model. Here we go. Maybe I could share my screen so you can see this. I'm going to screw everything up probably. There you go. This is my email. But look, this guy Mark at a company. What is it? And there's another one. I think the spike is too big and compared with the envelope, But that's what the spike looks like. There you go. It's cool, right? And let's see if I can get the camera back. There we go. It did it. Oh, my gosh. Dedicated studio space sounds expensive. I'm going to share it with Daniel Griffin. So that'll cut the cost in half. And but I do because I would just stay there all day and make content. And not just TWIVs, but videos to explain things. I've already thought of 5 different videos I want to make, like, SARS-CoV-2 won't cause infertility. It does explain variants, strains, and isolates, why hydroxychloroquine failed. I want to do all of that, and a studio would really help. Thanks, Tom, for your T. Klepper for your super chat. It'll come up eventually. I've learned that now. Is the UK correct in the delayed timing? We did one paper on TWIV, I think it was last week, where the delay looked good, but the numbers are pretty small. So they found that the longer you waited for the second boost, the better your antibody response was because you have time to mature the immune response. It just doesn't happen in 4 weeks completely. All right, let's try Claudia's. If a COVID-vaccinated person gets infected, how long does it take until antibodies disable the virus so that it can no longer replicate and be shedded? So in a healthy person that doesn't develop long COVID, that doesn't develop, say, inflammatory disease, about 2 weeks. But that's not the maximal antibody response. Amy would get very mad if I said this. It takes more weeks to develop, but you can start to clear infection in a couple of weeks. How does the virus clear from the body in 7 to 10 days if antibodies are not present? So other immune mechanisms are at play. In a healthy person, innate immunity could contribute to that. Other nonspecific immune cells. Here, let me show you a picture. Oh, that's Daniel Griffin. No. This one. This is Daniel Griffin's time course. And Amy won't let me show this. She doesn't like it. But here we go. Let me get rid of this comment here. Here we go. Here's time of exposure. And then the purple is viral load. And then you see it starts to go up. There's an incubation period before symptom onset, 2 to 14 days. Between 0 and 4 days, the viral load goes up. It peaks at symptom onset, that's TS, and then it starts to go down in the next week. And below these dotted lines, you're no longer infectious. So that's like 2 to 3 weeks. And you could have some antibodies, you could have some T cells, etc. doing that. Super chat from AB. Your chat will come up. I'll get to it. Super chat's supposed to go to the top. I don't know why it's just being put into the stream. Yeah, we can do playback speed changing. So for people who thought Paul Offit talked too quickly, you could slow it down. It's great. Okay, Mike, why shouldn't we get the J&J vaccine after completing the set of mRNA vaccine to further boost our immune system? So you're asking, can you just get infinite boosters? You don't need to. I don't think it further boosts your... I think the quality of antibodies and memory B cells and T cells are the same after 2. So it's not going to help. That's why. That's my thought. Thank you for your super chat, Tom. So these donations will go towards a studio in New York City. And then if any of you visit New York, you could come to the studio and visit. I think that's cool, right? Will adenovirus vectors for new variants be less effective in people who already received the original vector vaccine due to vector immunity? So this is a good question. And I originally thought this was an issue, but for some vectors, it apparently is not. And in the vaccine trials, it's been, we are seeing now that some adenovectors don't present an issue with respect to that. So I don't think it's necessarily, it's a problem. It really depends on the vector. Can virus be small enough for quantum tunneling? I don't know what that is. You know, the limit of my science is viruses and a little bacteria. And I know a lot about viruses, though, but I don't know about quantum tunneling. Clippy, thank you for your super chat. When in science history has the total understanding of human behavior changed in weeks before? I'm not a historian either. Maybe it did in the 1918 pandemic, and it's been forgotten by many people, right? Because that was a pretty substantial thing. I also think wars changed society a lot, especially families of people who were killed. So, but that's not science, I understand. You like this communication style where we do live streams and answer questions? I think it's great. It's like having office hours for my virology course, right? I love it. I do love it. I don't watch this show. No, I do not. Sorry. Mary, thank you for your super chat. What are your favorite side hobbies? Well, I don't know about Amy. I think her side hobby is science, and so is mine. Mine is communicating science. I really don't, I like to read, but I don't think that's a hobby, right? Basic question. For mRNA vaccines with 95% protection from severe disease, does it equally protect against infection? Or is chance of infection merely reduced? So that's a good question. We do not know. I think it's likely that they're going to reduce infection and reduce transmission. There's a study out of Israel now that suggests the Pfizer vaccine decreases shedding. And so I think we'll see more and more of that. And it's likely it's going to decrease shedding, which is a good thing for herd immunity, of course. What changes in this virus would worry you? So it's not like I'm not worried. I think variants are concerning because they could evade immunity, right? Although I find it hard to believe that they would completely evade vaccine-based immunity in a very short period of time. You know, we change flu vaccines every year or so. It's not a big deal. When the virus changes, you can get a mild illness with your previous immunity, but you don't get life-threatening illness. So I'm not concerned about these variants in that way. What would concern me? I don't see anything that concerns me because I understand how viruses behave. They don't change overnight to become more transmissible or completely evade immunity. It just doesn't work that way. And we know from our history. So I'm not going to say that there's anything that worries me. I would worry if we didn't have a vaccine by now, and we do. The like button is just down below the window. We have now 219 likes and 490 people. You guys are amazing that you come here and you want to chat about science. This is the coolest thing. Below the video. Yep. Thank you, Arthur, for your super chat. You got us past the 100 mark. Yep. Can the virus mutate in people who are already hosting the virus? Sure. As anybody the virus enters, gets in the person, it's mutating in every cell it's reproducing in. A lot of those mutations inactivate the virus, so we never see them. Some of them are neutral. They don't do anything. And so some of them may give new properties, like being resistant to monoclonal antibodies. So how many people in the U.S. are vaccinated? 55 million doses have been administered. That's 11.9% of the human population. I forgot to click that question from John in Ireland. So we're slowly getting there, and it's going to get quicker and quicker. Eugene, thank you for your super chat so much. And Claudia, thank you for your super chat. You guys know why the government lab my mother worked back in the 90s wanted many years for testing every vaccine? Well, it just, first of all, it wasn't an emergency, so they took their time. They would do a phase 1, then wait, and then do a phase 2 and wait, and a phase 3. And we're in an emergency, so we needed to develop a vaccine. But this should not be taken to say that we cut corners. We just did it quickly, and all the safety is in. It looks really good. And I asked Paul Offit, what's missing from the safety? He said nothing. It's all there. It's all good. Yes, Sharon, I also have a difficult time understanding the position of the CDC. If you're fully vaccinated, exposed, you don't have to quarantine. I don't think they have the data to say that. I don't know why they're saying it. I don't get it. Super chat from Make Mine Caffeined. Thank you so much. A pandemic or epidemic, don't give a political answer. This is a pandemic. It means the infection is throughout the world. If an epidemic is local, say if SARS-CoV-2 stayed in China, it would be an epidemic. It went to the rest of the world, it's a pandemic. That's all there is to it. It's a scientific definition. No more, no less. Yes, I did. She has cool 3D viruses in the lab. I forgot. And I'll get her to do it next time for sure. Absolutely. Why didn't they design vaccines against more regions than spike? So here was the idea. So the spike, here's the spike on my little model, my 3D printed model. The spike is what attaches to the cell, right? And that leads to the virus getting in. So the idea is that antibodies to the spike would block infection. And we know that for many viruses. So they decided to just focus on one protein. So the mRNA vaccine is the vectored vaccine. So when you do a vectored vaccine or an mRNA vaccine, you have to pick one. You could do two or so, but they decided spike would be it. And I questioned that early on. They were right. It looks good. It looks really good. Suzanne, thank you for your super chat. How are Amy's black assays? She does them every day. The lab is full of them. Plates stacked up like crazy. She had a bad day today because some of her cells crashed. She was not very happy. Vaccine progress has been phenomenal, but we need to continue with therapeutic developments. Absolutely. Absolutely. There are therapeutics in development, and they have gone more slowly, and they're not getting the same attention as the vaccines. But you'll hear more about them, I'm pretty sure. It's a community that appreciates real scientists and not fake media scientists and government agencies. So I guess you're talking about our little community. So we have a community here that we've made Wednesday nights, live streaming. Yeah, I think it's great. I'm a real scientist. I'm interested in science. A lot of people, say I have other agendas and so forth. I don't. I'm really just interested in science. I don't own stock in companies. I don't have any agenda here. So I'm just interested in science and talking about it with you. Are there any viruses that are not COVID that recombine with COVID that you're worried about? No, anything that's not a coronavirus, it's not going to recombine. It's too different. Only the coronaviruses. But, they can, there are a lot of coronas out there that this could recombine with, say, in a bat or some other animal. So we have to be on the lookout. Yep, 45 minutes went by. That was the Amy part, right? Yep. You think people won't still be concerned about this a few years on? I think they will be. I think in society, there'll be long-term effects of this. I think, though, after this year, the number of infections are going to go down. The deaths are going to go down. The vaccines will work. It will become another flu-like illness that we have to vaccinate against regularly. So I know you don't like it when Amy leaves, but, she's going back to work. Because she's still in the lab. Here it is 9/11 in New York. She went back to the lab. She's going to do more experiments probably till 10, 11 o'clock. Do either of you think that long COVID could be caused by persistent elevated immune surveillance? Yeah, I think so. I think an inflammatory process long term, and why that's happening, we don't know. Something's been perturbed by infection, but there's no virus around, and that's what has to be sorted out. Yes, data showed that B1117 had delayed clearance and not increased viral load. Yeah, I saw that. And what is it? A preprint? Yeah. So it's not on this week's TWIV agenda, but maybe next week. I'd like to look at that in some detail. Yeah. Thanks, A&B, for your super chat. Yeah, where else can you find a free virology class? That's right. Post-polio news. Thank you for your super chat. Post-polio news. Cool. I follow polio eradication, of course, because it's the virus I worked on for many years. Friend with lupus got COVID-19 twice, both times negative with nasal PCR. Only a blood test showed how she was infected. 0 antibodies. Well, yeah, some people never make antibodies. It's quite interesting. And that's why you still have to be vaccinated. Is it feasible that spike only could actually be more affected than one with more general epitopes? The immune system focuses. No, I don't think that you need that. You can give the immune system a lot of antigen. You can give them 100 different serotypes of rhinoviruses, and they deal with it. So I don't think that's the issue. I don't think that's the issue. Here's a super chat from Tom. Pleasure and privilege to visit you. Maybe have Eric Fried back. Yeah, we could have Eric Fried back. You bet. Are you able to read through all the comments after the live stream? I'm not sure if they stay or they go. Does anyone know? Uh, 229E, what does it stand for? It's just an isolate number. No, E is not E. coli. Nope. Julie, thank you for your super chat. Appreciate it. What changes should the CDC make to handle future pandemics? We have to do better surveillance of wildlife, the ones we think are the reservoirs for the next virus, bats, rodents, in all countries, not just China. The paper came out from Lin-Fa Wang, which we're going to do on Friday's TWIV. They find SARS-like coronaviruses in Southeast Asia. So we have to do more surveillance. and have a pandemic plan would also help. Thank you, Questella, for your super chat. Really appreciate it. Yeah, asymptomatic people can develop long COVID. Absolutely. I've heard, it's not published. I've heard people tell me that. What's the next pandemic? Influenza. And then after that, another coronavirus. Hit the likes. We're doing well on likes. I'm reading your chat here. It's interesting to just read it because you guys are chatting among yourselves, and that's cool. I do the same thing in my course, by the way. They have a chat among themselves, and I'm looking at it because it's interesting. Yes, Moderna has been perfecting their mRNA technology for a long time. Really good stuff. Thank you, Harry, for your super chat. No, I'm not sick. I got my second dose of vaccine yesterday, and I had a fever this morning, but I'm okay now. Periodically, I lose my voice because I talk all day. I taught today. I did office hours. I did a live stream and this. So I lose my voice. We have a vaccine for SARS-CoV 2003. No, we don't. A few were experimentally made, but they never got very far. And it's quite different. I mean, it's less homology than the bat virus, RATG, that we were talking about earlier, which is clearly a SARS-like coronavirus, but less similar than these bat viruses. Where do I draw questions from? There's a chat. It should be to the right of the video window. There's a chat there, and people can type in their questions, and I see them. I guess, Mrs. Anderson, that's what you're asking me, right? India's developing a nasal spray in phase 1. Why didn't we do this a year ago? I can't answer that. I wasn't privy to the discussions. I think it would have been a good idea to do that and diversify instead of having, well, the thing is, Pfizer and Mrna, they can't singly make enough doses to immunize everyone. So we need, Lynn, thank you for your super chat. We need more companies making it. And, you know, I think they thought that the mRNA was a real shot. So a sure thing, I should say. Thank you, T. Klepper, for your super chat. Really appreciated. What should we say to conspirators who believe the virus had never been isolated? Of course it's been isolated. They don't know. They haven't read the paper. The very first paper, which I go over in my virology course, by the way, they did a bronchial wash of a patient with pneumonia. So you put a tube in the lung. You wash with saline. You take it out. They looked at it under the electron microscope. Thank you, Plinky. Oh, this is very generous of you. Put it under the electron microscope. They saw coronavirus particles. They put it on cells in culture. They got the virus, and they sequenced the genome. I don't know what more you would want from that. They actually don't know what they're talking about. I hate to say it. Thank you, Les Fabi. Yeah, the name is familiar. Fan since your Twit Network days. Yeah. I used to be on the Twit Network. Yes, I'm going to make more explanatory videos. I will. And that'll keep us going. There'll be more virus. You know, there's currently an Ebola outbreak in Africa. Nobody is really paying attention to it, but it's killing people, right? Super chat from AB. Are there viruses that are highly communicable and dormant for weeks, months, then lethal? If so, given the response to government public, I'm worried. Well, HIV, you get infected, and then you can have years of incubation with mild disease, and then suddenly it destroys your T cells and it kills you. Yeah. So that's an example of that. Has there ever been a vaccine that was stopped because the vaccine was unsafe? Yeah. There have been a couple. Two come to mind, measles vaccine in the 60s and respiratory syncytial vaccine. They were both inactivated vaccines, and they were put into kids and they caused more severe disease. And so they were discontinued for sure. Yes, I've seen this preprint, increased delayed clearance and not increased load. I have to look at it. I haven't looked at it, just saw it today. And thanks, Jake, for your super chat. I'm going to read it for next week's TWIV. I haven't done it yet. We can talk about it next week. All right, Celtic, take care. Good night. There are some medicinal plants that are antiviral, for sure. There are lots of antivirals, all kinds of anti-compounds in plants. And ivermectin came from a fungus originally, a Streptomyces. Thank you, Clippy, for your super chat. Yeah, I'm very excited about the studio, yeah. And I'm limited by here in my basement. This is okay, but I want to have people helping me, and I need a bigger studio, and my office isn't sufficient either. So I'm looking forward to that. Why is there need for a vaccine? Because the reason the decline is probably because we're vaccinating people. And there are going to be future outbreaks if we don't have a vaccine. That's why we need it. Am I convinced of higher transmission? So here's my take. Transmission means you exhale a droplet, it goes through the air with virus, and it gets to someone else. So they're saying that's better. And that's not what happens, because B117 went to California and it has not taken over there. So it's not just more airborne transmissible. It may be more fit in certain situations like the UK. It seems to be taking over in Florida. Okay, but it's not transmission. It's something else that's going on, and it hasn't been figured out. What do you think of backward transmission from humans to animals may propagate reinfections? Yeah, I mean, the problem is if it goes into animals, it can sustain other changes. Thank you, Susan, for your generous super chat. Before I end up, I'll try and whip through the super chats really quickly, because I appreciate your contributions to our future studio, I suppose. Animals can harbor the virus and they can change, and some of the mink variants that evolved could, avoid antibodies. So that's a real concern, and we need to be really careful. How does it get in urine? So there's some idea that it enters in it's in the blood. Small amounts are in the blood. So, of course, that gets filtered in the kidney, and so that's how it could get there. And, it's RNA, as far as we know, not an infectious virus. Can you super chat Bitcoin? I have no idea. Okay, here's tunneling. A quantum mechanical phenomenon when a particle is able to penetrate through an energy barrier that is higher in energy than the particle's kinetic energy. Sounds like the virus is too big for that. What will the world look like in 5 years? Yeah, I think the pandemic will be over by the end of this year for sure. And we go back to schools and restaurants and all that stuff that you like to do. This virus is going to stay around, though. And we're going to have to either vaccinate against it, or hopefully it becomes a mild common cold coronavirus. If new mRNA vaccines are developed, would people who are already afflicted with recurrent HSV, HIV, or HPV get vaccinated? I don't think it's going to help those individuals, because those viruses are there, stuck there. Guitar, I haven't picked that up in years. I used to try and teach myself, and I got reasonably good, but I stopped, and I'm just thinking about viruses now. I do this podcasting stuff a lot. Okay, so we have 5-6 minutes left. Let's see for some super chats here. Everyone makes variants. Whether they mean anything is the question. Thank you, Make Mine Caffeinated. It's my pleasure to do this. Let's find the next super chat. Suzanne, thank you, Suzanne. I don't know if you guys are still here. DR, I realize you're a virologist. However, I'm wondering if you have thoughts on the risk of a bacterial pandemic emerging, especially giving antibiotic overuse. Well, yeah, we could have, for example, a tuberculosis pandemic where there's resistance to all the drugs we have, and it causes a global outbreak. That's very likely, and other bacteria could be an issue as well. AB Super Chat, why so many conflicting opinions on masking? Were they really so little definitive data on mask efficacy? In Asia, masking was a given. Yeah, well, problem is it's not easy to test masks, and so there were conflicting results. Some studies said they were good, some studies said they weren't, so people who didn't want a mask took on the studies that weren't so positive, and they say this shows that they're no good. And meanwhile, yes, in Asia, people wear masks, and that seems to help. So, you know, in New York, most people are pretty good about masks, but I know that in other parts of the country, they're not so good. But, you know, it's not a big deal. I go out with a mask all the time. Thank you, Lynn, for your super chat. Plinky, thank you for your super chat. Really appreciated. San Francisco. I'm going to look for the super chats and thank them right now. Thank Les. Yeah, you've been around a while. Twit, This Week in Tech. That's what gave me the idea for This Week in Virology. Thanks, Wendy, for your super chat. Sound is excellent. I tell you, I really put a lot of time into fixing this sound. I spent hours down here in my office, and I made some changes that I should have made a long time ago, and I think we got it a little better now, and we're going to make Amy's better. I know some people feel she has a high voice and it's louder, so I actually bought her an equalizer, and I'm going to tune her frequencies in. I'm out of my mind. That may be my hobby, you know, doing audio and video. I don't know. Jake, thank you for your super chat. Can I talk more about variants? So the media is overblown. They take scientists who say they're more transmissible and more virulent, and they go with that. But there are just as many scientists who say it hasn't been proven. I have lots of colleagues who say it wasn't been proven. The only thing you should worry about is not transmissibility, is whether these variants will escape vaccine-generated antibodies. And so far, it looks okay. The vaccines are still going to work, but we have to keep an eye on it. We do need to monitor these variants more carefully. We need to do a lot of sequencing. I think that people will be trying HIV mRNA vaccines for sure. Absolutely for sure. Thank you, Susan, for your super chat. It's really nice of you. You guys are amazing. You guys are just great. And I didn't even, think of super chat. It's just, and this week up in my YouTube channel, the thing came up, say, oh, you're eligible for super chat. So I clicked it, and now you guys are actually using it. That's really cool. I'm scrolling through all the super chats. So before I leave, here, Ariel super chat. Thank you so much. It is my pleasure to educate all of you. It is absolutely my pleasure. I might be interested in Mark Lipsich's perspective on challenge trials. I'll take a look. I'm not a fan of Mark Lipsich, I have to tell you. We've had our collisions in the past over gain-of-function research. I tend to be outspoken. I'm cranky. What is it? What is it that they call me? Grumpy, right? I speak my mind. I think it's important that scientists do and not be quiet. Has SARS-CoV-2 been fully genetically sequenced? Absolutely. Hundreds, thousands of times. Thousands of times. I'm running a compressor and an expander on this audio. It's a software-based one. I have actually a hardware expander compressor right here, but I haven't hooked it up yet. I'm working on it. But there is a compressor here, but I do very light compression, and I have an expander because there's some hissing noise here in the room. Thank you, Tensi, for your super chat. Thank you, Couch. Here's a bit of money for your possible new hobby. Yeah. Educating the world. I think you need to have good audio. I worked really hard to do that. I work hard to get good video. And so I don't want crappy audio. And that's why earlier in these live streams, people said I had crappy audio. I got on it right away. Yes, I will do my best to continue at its current frequency. I do have to have co-hosts join me. And it depends on them, too. I will stay grumpy. I will absolutely stay grumpy. Yeah, I don't know what's going on with this Brazil variant, okay, the second wave. There's some issues. I read the paper today, and they're not even sure what's going on. It may not just be the variant, so stay tuned. Thank you, Stephen, for your super chat. Thank you, Dylan. Dylan-iated. I don't know how to say it. Am I cantankerous or curmudgeon-y? I don't know about curmudgeon. I'm grumpy because I want people to get things right, and I want them to think about things. You haven't heard me about gain of function. So this was like 8 years ago. I got onto that, and we talked a lot about it on Twiv. I blogged a lot about it. I went to meetings and talked about it. I don't think you're grumpy at all. You just don't take crap when challenged and will snap at nonsense. I love it. Thank you very much. Thank you. Some people don't like it. That's why I have 4 thumbs down here, because they don't like it. Oh, well. When will COVID become a new strain, not a new variant? Maybe never. You have to have a really different property. There are no strains of HIV-1. I asked an HIV virologist. And he said there's no strains. There's just variants. So you'd have to have something really different. Really different. Thank you, Overjoyed, for your super chat. And super sticker, actually. That's cool. Do I agree with Amy that the case drop is not real? I don't. And tomorrow she's going to listen to this and get mad at me when I go in. But I think the hospitalization drop is some indication that there is an actual drop in cases. Suzanne, thank you for your super sticker. That's cute. They described the super stickers. All right, let's wrap this up. It's 9:32. I could stay a long time, but I don't want to keep you guys here forever. Then you get tired of it. Should we be scared about the variants being resistant to the vaccines? No, don't be scared. I think we can handle it. I think so far they look good. And if infections go down, you're going to have fewer variants emerging. So, it's a matter of getting people immunized. So I think that's great. All right, folks. I think that's it for tonight. Let me thank you all for being here. It's great. Your questions are great. And we'll see you next week. What kind of mic? This is a Rodecaster mic. It's $99 mic. I used to use a $300 mic in the studio for years on Twiv. And when I came home, I had to get a mic here, and I just spent $99 on this. I put a windscreen on it, which really helps. Anyway, folks, thanks again, and good night. See you next time.